Welcome to this lecture on the nuclear receptor pathway. Imagine a highly exclusive nightclub where most people have to pass messages through a strict bouncer at the front door. This is exactly how most cell signals work, stopping at the cell membrane. But what if a very important VIP arrives? The VIP ignores the bouncer, walks right through the walls, and goes straight to the manager's office to change the music. In our bodies, these VIPs are specific hormones, and the manager's office is the cell nucleus. Today, we will explore this special VIP route that allows unique signals to directly alter how the cell behaves. Now that we have introduced the topic, let us start with a helpful analogy. Imagine a highly exclusive nightclub where most people have to pass messages through a strict bouncer at the front door. This is exactly how most cell signals work, stopping at the cell membrane and passing a message inside. But what if a very important VIP arrives at the club? The VIP ignores the bouncer, walks right through the walls, and goes straight to the manager's office to change the music. In our bodies, these VIPs are specific hormones, and the manager's office is the cell nucleus. Moving from our analogy to actual biology, let us look at the molecules that act as these VIPs. In molecular biology, the nuclear receptor pathway is a specialized signaling mechanism. It is exclusively used by small, hydrophobic, or water-fearing molecules. These include steroid hormones, thyroid hormones, and certain vitamins. Because these molecules are lipid-soluble, they possess a unique physical property. They can easily diffuse directly across the lipid bilayer of the cell membrane. They do not need to wait outside and bind to a surface receptor like other water-soluble molecules do. They simply pass right through the cellular wall. Now that these hormones are inside the cell, what happens next? Once inside the cytoplasm or the nucleus, they seek out their specific partners. These partners are intracellular proteins called nuclear receptors. Upon binding the ligand, the nuclear receptor undergoes a dramatic physical shape change. This conformational shift is absolutely critical. It transforms the receptor from an inactive waiting protein into an active transcription factor. It is now a fully functional molecular machine, primed and ready to interact directly with the cell's genetic code to make sweeping changes. With the receptor now activated, we can understand how it actually changes the cell's behavior. This activated hormone receptor complex travels directly to the DNA. Once there, it binds to very specific DNA sequences known as hormone response elements. By binding to these specific regions, the complex acts exactly like a light switch. It can turn target genes on, initiating the production of new proteins, or it can turn them off, halting production. This direct interaction with DNA is what makes the nuclear receptor pathway so powerful and efficient at altering cellular function. Why does this direct gene control matter so much to our survival? It is the fundamental mechanism behind how our bodies regulate growth, development, metabolism, and reproduction. For example, sex hormones like estrogen and testosterone use this exact pathway to trigger the massive physical changes associated with puberty. In the medical world, understanding this pathway is absolutely critical for treating severe diseases like breast cancer or prostate cancer. Doctors use specialized drugs that specifically block these nuclear receptors. By doing so, they prevent the cancer cells from receiving the growth signals they desperately need to multiply and spread. Before we move on to the mathematics of this process, let us visualize the entire biological journey we just discussed. Here you can see the cell membrane at the top. Notice how the steroid hormone, represented by the hexagon, easily passes right through the lipid bilayer into the cytoplasm. Once inside, it finds its specific nuclear receptor and binds to it, creating the hormone receptor complex. Follow the arrows, and you will see this complex traveling through the nuclear pore, crossing the nuclear envelope to enter the nucleus. Finally, it binds directly to the DNA at the hormone response element. This binding event triggers gene transcription, producing the mRNA you see at the bottom. This diagram perfectly summarizes the direct control these hormones have over our genes. Now that we understand the biological mechanism, let us look at how scientists measure this activation mathematically. To understand how strongly a hormone activates this pathway, we must look at the binding affinity between the hormone and the nuclear receptor. We express the fraction of bound receptors using the standard fractional occupancy equation. In this formula, Y represents the fractional occupancy, which is the proportion of receptors currently bound to a hormone. The symbol L in brackets represents the concentration of the free ligand, or hormone, inside the cell. Kd is the dissociation constant. It represents the ligand concentration at which exactly half of the receptors are occupied. If the ligand concentration increases, Y approaches 1, meaning all receptors are bound. If Kd is very high, the receptor has a low affinity, requiring much more hormone to activate the pathway. Let us apply this formula to a real biological scenario to see how it works in practice. Imagine a cell exposed to the stress hormone cortisol during a fight-or-flight response. We are given that the intracellular concentration of cortisol is 40 nanomolar, and the glucocorticoid receptor has a dissociation constant of 10 nanomolar. We need to calculate the fractional occupancy to determine the strength of the stress response. We start by substituting our known values into the equation, placing 40 in the numerator and both 10 and 40 in the denominator. Adding the denominator gives us 40 divided by 50. Simplifying this fraction gives us a final answer of 0.80. Physically, this means exactly 80% of the receptors are bound to cortisol, indicating a very strong and rapid stress response. Having seen how to calculate occupancy, we now face a different challenge. What if we know the desired occupancy, but need to find the required drug dose? A pharmaceutical scientist is designing a synthetic hormone with a dissociation constant of 5 nanomolar. They need to achieve a 90% receptor occupancy. We must rearrange our equation algebraically to solve for the unknown ligand concentration. This is a crucial step in pharmacology to ensure correct dosing without toxicity. We start by plugging in 0.90 for Y and 5 for Kd. To isolate the unknown variable, our first mathematical step is to multiply both sides by the entire denominator. This eliminates the fraction and brings the unknown variable up to the main line of the equation, making it much easier to work with. Continuing from our previous step, we now need to expand the expression on the left side. We distribute the 0.90 into the parentheses. Multiplying it by 5 gives us 4.5, and multiplying it by the ligand concentration gives us our second term. Now we need to group all terms containing our unknown variable on one side of the equal sign. We do this by subtracting 0.90 times L from the left side and moving it to the right. The constant value of 4.5 remains isolated on the left. Finally, we subtract the decimal from the invisible coefficient of 1 on the right side. This simplifies to a clean coefficient of 0.10. Our equation is now in its simplest linear form. We have reached the final stage of our derivation. To completely isolate the unknown variable, we perform one final division step. We divide the constant, 4.5, by the decimal coefficient, 0.10. The result is exactly 45 nanomolar. This mathematical proof tells the pharmaceutical scientist that the intracellular concentration of the synthetic hormone must be maintained at exactly 45 nanomolar. Achieving this specific concentration ensures the desired 90% activation of the nuclear receptor pathway. This proves how algebraic manipulation of the fractional occupancy equation directly informs real-world medical treatments and drug design. We have covered a lot of ground today, so let us summarize the most important ideas. First, the nuclear receptor pathway is an elegant method of direct cellular communication used by lipid-soluble molecules. By bypassing surface receptors, these hormones bind directly to intracellular transcription factors, exerting profound control over gene expression. Second, we learned that the biological mechanism is deeply connected to mathematical binding kinetics. The fractional occupancy equation allows us to predict exactly how a cell will respond to different hormone doses. Finally, understanding both the biology and the math is essential for modern medicine, allowing us to design life-saving pharmacological treatments. You now have a solid understanding of how these VIP molecules control our cells from the inside out.